The overall goal of this procedure is to visualize Nanopodia through immunofluorescence staining in cultured cells in vitro. This is accomplished by first culturing the cells of interest on collagen-coated glass discs. In the second step, the cells are fixed to the disc with warm PFA. Next, the cells are washed and then incubated in blocking buffer for 1 hour at room temperature. In the final step, the cells are stained with antibody against TM4-SF1 and mounted on a glass slide. Ultimately, the presence of Nanopodia extensions in mobile, proliferating, or interacting cells can be visualized by immunofluorescence microscopy.
The implications of this technique extend toward the therapy and diagnosis of human cancers, because endothelial cell and tumor cell proliferation, movement, and intercellular interactions are key processes involved in the angiogenesis and growth of tumor cells. Demonstrating the procedure will be Chi-Iou Lin, a postdoc from my laboratory.
Begin by autoclaving 12 mm glass discs in a 4-ounce glass jar. While the discs are being sterilized, place a 50 ml conical tube containing 25 ml of 70% ethanol into a cell culture hood. Place a sharp forceps with an extra fine point into the ethanol for 5 minutes, and then carefully remove the forceps and cap the tube, placing the forceps on top of the tube to air dry for 2 minutes.